everyone, and welcome to the Synthace DOE Masterclass. My name is Khaled Elwad. I'm a scientific solutions consultant here at Synthace, and I'll be your host for today's session. Before we start the presentation, I'd like to just explain the format and some of the features that will help you get the most out of this session. Please make sure that your Zoom settings are set to computer audio only, and should you have any technical difficulties, send us a private message in the chat box below. The webinar is being recorded and will be emailed to you afterwards, as well as being made available on our website, so you can watch it on demand. But we encourage you to take advantage of this interactive session and ask questions into the Q&A box at the bottom of your screen. We'll be able to answer these questions during the second half of the webinar, but please feel free to add them to the Q&A box at any time during the presentation. So there's a huge amount of depth and detail to DOE, and as we won't be able to cover it all in one session, our first session will give you an introduction to DOE and show you how power that it has to transform your experimentation. And in the second session, we'll walk through detailed real-life examples of where we've used DOE, and the third session will show you how to perform a DOE in a pragmatic way. By the end of this three-part intro series, you'll have the starting tools you need to run your own DOE. So if you haven't already done so, please sign up for the second and third webinars. The link to do so is going to be in the chat box below. Keep an eye out for future webinars in this series as well. Now I'm excited to introduce our presenter for today's webinar, Markus Gershater, Chief Scientific Officer and co-founder of Synthace. Markus has been applying DOE to biological problems for over 15 years in both industry and academia. His conviction that biology can be transformed by multifactorial methods was the main driving force for him to co-found Synthace. And Markus's diverse background has given him a very broad range of expertise covering biochemistry, process development, assay development, and much more. So now, without further ado, we'll get started and I'll hand it over to Markus Gershater.
Thanks a lot, Khaled. And thank you all for joining us today. I have to say, it's really exciting to see the responses series has received. We've had over 900 registrants, which is absolutely amazing. Having been passionate about using DOE at Synthace since we started, it's great to share some of that experience with you. As Khaled alluded to, teaching DOE in full is well beyond the scope of a single webinar, and actually in full is well beyond the scope of this introductory series. But that's essentially what we're looking to do over the next 3 webinars. My job in this introductory webinar is to introduce you to DOE for biology and to really show you its power. So, as a really passionate advocate for this kind of methodology, what I want to convince you of is DOE's transformative potential for the work that you're doing. So I kind of wanted to see how big a job I've got to do. And so for that, we've got a quick poll that we'd like you to fill out and just give us an idea as to how bought in to DOE you already are. So just while that's filling out, I mean, yeah, like I say, it's been really exciting to have so many of you with us today. In a lot of ways, it's quite sad it's virtual, because if we were in person, then this would need to be in a really big lecture theatre and we'd all have a real visceral sense as to this surge of interest that we've seen in multifactorial methods. Just scrolling through the list of all the attendees has been really quite inspirational. As we're a company that's been had this at our core for a very long time. So it's yeah, really exciting to see how switched on to DOE the domain's becoming. So hopefully that's given you all a chance to vote, and be interested to see what the results look like. Okay, so it looks like yeah, there's some people. Most people are saying they've heard of DOE and it sounds potentially of use. We've also got a decent number, closely followed by the decent number of fanatics, which is fantastic to see as well. I don't know whether I'll be able to bring you much new to those of you who are already really convinced by DOE, but hopefully it'll be enjoyable for you nonetheless. And for those of you who haven't got an opinion yet, or want to hear a little bit more about it, well, that's exactly what this session's about. And we'll run a similar poll towards the end so we can see how we've done. Let's get going. So first off, I want to start thinking about some high-level concepts. So first off, I think we can start with stuff which should be pretty uncontroversial and something we can all agree on. Doing biology is tough. It's really not easy, and the experiments we use to try and get an insight into our biological systems that we're working on are really costly. So fundamentally, this is why you're all here. If running experiments was cheap and easy, then we could pretty much just brute force our way through to the answers we need. But actually, the vast majority of experiments are not cheap, they're not easy. And so we need to think about how we're doing them. So there's this fundamental question of how should we experiment? And where I really want to start is how are most experiments conducted today? And this is actually something which in the UK is taught as running fair tests. So a sort of GCSE level. So this is kind of when you're 14, 15, 16, then you're taught this concept that you have to hold everything in your system constant except the one thing that you're investigating. Otherwise, how do you know whether it's that thing that's having the effect that you're observing or not? And this is also known as one factor at a time. So let's just run through this for a second. So let's say we've got our biological system and we want to start investigating it. We pick a factor that's likely to have an impact on that system, or most likely to have an impact on that system, say temperature. And we think about the range that we want to look at temperature over. We pick levels within that range, and we run experiments where each one of these dots represents an experiment in the or a run in that experiment, or a set of runs, because we'd obviously run it replicated so that we could see how good our results were and what sort of signal to noise we were seeing. So we'd run the experiments, we'd take our replicates, we'd average them, and we'd plot them on a graph, and we'd get some kind of a response to that factor, draw a line through it. So then we think, okay, well, we found the best temperature. We now want to look at another factor. So we might say, look at pH, and think of different levels that we might want to look at pH. Now we'll obviously do this at 25 degrees because that was our best temperature. And we run another set of experiments and we draw another line through it. And we say, okay, this is our optimum. The issue with this, and as hopefully is made reasonably clear by plotting out the experiments in this way, we've actually made an assumption about a two-dimensional design space, and we've just taken two cross sections through that design space. So we haven't really covered the overall area that effectively at all. And what this could mean is that if the optimal pH depends on temperature and vice versa, then actually what our system could actually look like would be something like this, and that would be entirely consistent with the results that we'd got from our two one factor at a time experiments. So our optimum could be somewhere over here, and we just wouldn't know. So this is a fundamental issue with one factor at a time experiments when we're in a system that might have interactions. So the next thing we need to think about is the nature of nature itself, right? So what is the system that we're working on? Fundamentally, biology is an emergent phenomenon. So every living thing is the result of multitudes of different parts, all interacting in an untold number of unpredictable ways. So, whether we think about cells or proteins, every protein's function is ultimately an emergent property of the sequence of its amino acids. Every system you'll work on as a biologist is complex in this way. So biology at its heart is all about interactions, but we're using methods that are blind to those interactions. And for me, this is a really fundamental point. We're all taught to do science in a particular way. We apply this, very diligently to our biological experiments, and yet it's missing one of the fundamental features of this system that has evolved over 4 billion years. So what's the alternative? Well, we're going to be talking today about multifactorial experimentation in the form of DOE, and we regard this as a really powerful tool to start going into more sophisticated experimentation. So an alternative experimental design could look something like this. So instead of just taking these cross sections through design space, we're putting a framework of experimental runs into that design space so that we can actually see how the system changes as we explore in a multi-dimensional way. So just to make this a little bit more plain, let's put some factor labels on here so we could imagine that this is temperature, time, and pH in this fictitious experimental system. And so you can see that for each one of these, we've looked at two levels. So low temperature, high temperature, low time, high time, and so on. And so what we need to do as scientists then is we need to think about, okay, what levels are actually relevant for us to test? So we go to all of our knowledge, we go to every bit of information we can possibly find out about our system, and we populate this design with specific levels that we might want to look at. So we're going to look from 25 degrees centigrade to 37 degrees centigrade, because those are relevant temperatures for the biological system we're working on. Leave it for 3 hours or leave it overnight. You know, these are fairly common levels that we might look at as biologists. And the point is, so if you want to see the effect of temperature, well, you look at the left-hand side of the cube and you compare it with the right-hand side of the cube. So, you know, at its heart, DOE is pretty simple. All it's doing is, you know, essentially looking at trends in these factors in a fairly simple way. It's fairly sort of simple linear interpolation, to put a slightly more formal term on it. But there's a really critical point here, because if we want to see what the effect of temperature is if we leave it overnight versus just for 3 hours, then we can also see that. So this is the way that these designs can start seeing how these factors interact with each other. You know, there's a huge number, as I've already said, there's a huge number of deep interactions within biology, and we need methods to help unpick that. But actually, this is just a conceptual experiment that you may run if you're looking at just 3 factors. But actually, DOE is a lot more powerful than that. We can look at a very large number of factors simultaneously. So this gives us an immense power to look at many, many hypotheses in parallel and really get a sense for our biology very, very rapidly by parallelizing this hypothesis testing. But there's a bit of an issue in doing that, in that, you know, as we add factors, then the number of experiments that we need to do to address those factors could go up exponentially. So this diagram here is essentially my effort at trying to show a 6-dimensional space in 2 dimensions, which is a little bit difficult. But essentially, we've still got temperature, time, and pH, but then we've added another 3 factors of A, B, and C on the corner of each one of these design spaces. So we're kind of going into this kind of hypercube. It doesn't matter exactly what the diagram means, but suffice to say that obviously the number of runs that we're running is going up dramatically. So this is a phenomenon that's known as an exponential explosion. So as the number of factors is going up, then the number of runs actually goes up by 2 to the n, where n is the number of factors you're looking at. So it's going up in a extremely fast and exponential manner. And that means that very quickly, your experiments would become completely unfeasible, even if you had a very high throughput system that you could work with. So in this case, we'd be looking at 6 factors with 64 experiments, where every single one of those experiments is different and varied over those 64 factors. So already over those 6 factors. So already it's a really complex experiment to run. But the good news is actually at the heart of design of experiments, another way that it's referred to is optimal experimental design. And it's about being quite disciplined about what we're asking with each experiment. And so we can use the principles of DOE to actually strip away a lot of these experimental data points and still gain the fundamental knowledge about how this system's working and some of the key interactions that are at the heart of this system. You know, for instance, if we want to see what temperature is doing, we can still compare one side of the cube with the other side of the cube. So this is just a very, very high level introduction as to the fundamental concepts as how we can explore multifactorial space by putting these sophisticated matrices of run conditions across the experimental space to really understand what's happening as we move through it. Now, that's about as much as I'm going to do on dry theory. I now just want to jump straight into an example so that you can get an idea of what this looks like in practice. So the example I'm going to take you through today is actually one that's from relatively early on in the company. And we were using DOE to optimize processes for producing mammalian proteins, so mammalian liver proteins. And this is actually an example of producing carboxylesterase 1, which is a drug metabolizing enzyme in the liver. And I'm going to use it as a way of kind of illustrating the overall DOE process. So that DOE process is broken down into 5 fundamental parts. The first part is something that we all do as biologists. You know, every time we design any experiment, whether that's a formal DOE or whether it's just, you know, whatever experiment we're running. And that is, we think about what we already know about the system, and we think about what we want to find out about the system. And we think about, okay, well, how are we going to lay the experiment out accordingly? Now, in terms of DOE, this is identifying the factors that we want to investigate. So you brainstorm every single factor you might want to investigate, and it's quite easy to get into the hundreds when you're getting really comprehensive about all of the different factors that might affect your process. And then you think about the levels that you might want to look at before, as I alluded to before when we were going through our fictitious example. But yeah, it's fundamentally about what do we already know and what do we want to find out, and you kind of sit your experiment in that space of the unknown, bounded by all the stuff that we already know. And so then when you've worked out that's all that kind of biological context, if you like, we then choose a design. So this is the bit that's kind of more specific to DOE. And it's kind of constrained by a number of different things. So we need to think about the practicalities of, just how much resource do we have to run this experiment? Can we run 8 runs of an experiment? Maybe if it's like a stirred tank bioreactor situation where you've got very expensive runs and you can just do 8 of them. Or you might be able to run 1536 runs if you're doing, some kind of high throughput experiment for assay development. And so, that will really then affect the experimental design that you might choose. And we want a design that's going to help us with what we want to find out, whilst making the most of what we already know. That's one of the key things in making a really elegant experimental design, is to make sure that we're not going over old ground too much. And then we also have to think about, well, once we've chosen a design for what we're going to do, we then actually have to plan out all of the details as to how we're going to run each one of those runs that the design tells us we need to do. So that's kind of all the design and planning stage. And then you actually go and run the experiment. So you get in the lab, you set up each one of the runs according to these matrix of conditions that the design of experiments design has dictated. You then need to gather all the data from the different analytics that you've used as part of that experiment and put it back in the context of the design. So you structure it back into the context of the design. And then you can build relatively simple empirical models from that, which tell you which factors are having an impact on your system and what effect they're having. And then you can look at those models and see how they fit with what you know. So at every stage, what we're doing is we're kind of applying our knowledge of biology and thinking about, what experiment we want to run, and then when we get the results, how those results relate to what we already know. And if we think about it, actually, pretty much all of this is how we'd run experiments normally. There's just a bit of a difference with how we'd choose a design and in the kind of planning stage accordingly, and then how we build models and draw conclusions, in that we have a much more powerful ability to derive insight from this kind of experiment than we might be used to. But ultimately, it's very similar to how we'd run things in general. So when we're choosing a design, it's often for a very specific purpose. Again, to get to really elegant experimental design, we want to be very clear about what we're doing with each step. So in the example that I'm now going to go through, we had 4 steps. So the first step was just to see which factors are important. We were actually investigating 15 factors in this experiment. And when we started, we didn't know which ones were actually going to have a big impact on the system or not. It's very easy in retrospect to look back and think, well, of course it was those factors. But we'd given it a huge amount of thought, a huge amount of literature searching and investigation in order to try and identify those factors. And we honestly couldn't tell you which ones would have an effect and which ones wouldn't a priori. So we used DOE to empirically test this. And because we're asking such a simple question, we can actually investigate 15 factors with just 64 experiments. And if we think, that sounds like, there should be something inherently wrong there, because if we were going to cover all of the potential design points within that space, so it would be 2 to the 15, which I think is something like 32,000 experiments. So we're reducing those 32,000 experiments to 64 experiments, and you would really think it wouldn't work. But actually, the mathematical principles that DOE is based on still hold out really well. So then what we found actually is that the bit of design space that we're in wasn't the bit that we needed to be in, and I'll get onto why that was in a minute. But what we did is we iterated. So we moved the design space that we were looking at to a slightly adjacent design space to get more data. And then by that point, we'd actually looked at these 15 factors from all sorts of different directions and pulled them in many different directions. And we found that 8 of them had just done nothing. we'd given them their chance, if you like, and they'd done absolutely nothing at all. So we focused in then all of our experimental power on just the 7 factors, which we'd found actually had quite a strong impact on our system. So we did another experiment just on those 7 factors, and you can get a lot more dense about those, and you can actually start to really define how those factors are interacting with each other at quite a deep level when you have this kind of density of design. And additionally, at this point, you might be able to see we've got an additional set of data points in the center of the design as well. So this is the first time we can actually start to look at curvature in the system. So we're not just looking at, which factors are having an effect and which way we need to go to get to a better system, but we're also starting to see whether there's something a little bit more subtle going on as well. And then finally, what we can do is we can take the 4 factors which have been most important throughout all of this, and we do a really augmented design where we've got 5 levels now for each of those factors. And so we can start modeling quite sophisticated response surfaces in there, as opposed to the simpler models that we had with these earlier screening type designs. So hopefully that can give you a sense of how we really answer very specific questions with each stage of the DOE, and it illustrates a fundamental strategy within DOE, and that's of iteration. You learn something, you build a model, you then use that information to then design your next experiment until you get to a place where you're satisfied. So our factor screen, like I say, was 15 factors, 64 experiments in 1 week. So that was the overall experimental strategy. And the data we got from it were these. So we were measuring activity of the enzyme because we weren't just interested in whether the enzyme was expressing or not. We wanted to know that it was active. So we're actually measuring the activity. And as you can see, there were an awful lot of conditions that yielded nothing, about 3/4 of them. But there were some of those conditions which we were really quite pleased with because we were getting quite a strong signal. We were getting quite a lot of expression. And this means that there's a decent amount for the stats to kind of get its teeth into, if you like, and actually build a model. So the modeling looks a bit like this. So I'm not going to go into detail in this webinar as to how these plots are made, but suffice to say, everything on this line is noise, and then everything off the line represents more significant factors, factors that are actually having an effect on the system. And DOE is often quite pragmatic. So, what we're going to look at are these factors that are most significant. They're furthest away from the line, furthest away from just being random noise. So we have here the factor labeled G, the factor labeled E, and we also have EG. So G was the co-expression of molecular chaperones. So these are proteins that help other proteins be produced. And then E was the amount of time that we were leaving the system to actually express the protein. So you need to leave it for a decent amount of time, and you need to have chaperones in place. But actually, it's more than just this. It wasn't just those 2 things which then cumulatively gave a response. It was there's actually a 2-factor interaction between E and G. So it meant that you only actually saw expression when you had the correct strain, i.e. the one that was co-expressing molecular chaperones, and you left it for enough time. So there's this synergy between these 2 factors. It's the only 2-factor interaction, or the only factor interactions that we actually have a word for. It's a synergy. People often overuse the word synergy, but this is actually what it is. So it explains the previous results that we had of not actually, the vast majority of the experiments didn't work, and that's because 3/4 of the design space didn't actually satisfy the constraints of having both molecular chaperones and being given enough time. So already, right in this beginning factor screen, we were seeing a fundamental interaction that if we'd been looking at 1 factor at a time, we could have completely missed this and had just assumed that we couldn't have expressed this protein. So the reason we then wanted to iterate was, because these 2 factors were so important. So we shifted the production time to being all overnight, basically. So previously we'd been looking for leaving it for 3 hours or overnight, and we realized that leaving it overnight was really important. So we moved the whole design space to being overnight, and we made sure there were molecular chaperones in place, just based off the fairly simple models that we produced. And the proof of the pudding's in the eating. And we ran the next experiment, and boom, everything goes up. And so we were already relatively pleased that we'd got some kind of signal here. And then just in, the next week, we'd already more than doubled it. And that was just by following the models that DOE had given us. It wasn't by doing anything particularly fancy. It was just by running this ordered set of experiments and following where it led. And the nice thing about it is obviously then we get a lot more data. You can see there's a reasonable spread in that data, which suggests that there are factors at play. And so then again, we can do some more modeling and we can see which factors are having which effect. And so we're seeing all sorts of other factors coming into play. As you can see, we were actually looking at all sorts of different types of factor in this experiment. We were looking at genetic factors, so how we were actually setting up the expression cassette for making this. Obviously, I've already mentioned the co-expression of chaperones, and we were looking at lots of different growth media factors, timing factors, temperature factors. And in this way, we could get this very holistic view of what was happening in this complex system very, very rapidly. And so we have all of these other insights, which we can then get the benefit of when we move to the next iteration, which was then where we really wanted to zoom into these factors that were having the biggest effect. So at this point, like I said, when I was going through the slide which summarized the overall experimental strategy, we were looking at really zooming in on the 7 factors which were most important. And it's actually kind of interesting here as well, though, because you know, you can use your biological knowledge and put it into these designs at pretty much every stage. So if we were thinking, okay, well, we're elongating the time, you know, as we're shifting through design space in this way. So maybe even though some growth some growth media factors weren't important before, they might be becoming important. So the modeling might not necessarily highlight them, but our biological knowledge still says, okay, we still need to actually look at these factors a little bit more. So it's not like you're just devolving all of your thought and all of your thought process to the statistics. It's really important that as biologists, we're looking at them in a critical way and iterating through things in a very considered way, as opposed to just blindly following it. And so, yeah, we looked in detail at these 7 factors with an additional 80 experiments, again, using just a week to get these further insights. And once again, the overall expression levels went up quite substantially. But the eagle-eyed amongst you will notice that actually we've got some zeros again, which isn't actually something we expected at this point. We thought we'd left zeros behind in the previous iteration. And so we knew that there was something, you know, potentially quite interesting going on here. And in the analysis, what it came down to was a 3-factor interaction, right? So 2-factor interactions we've already seen an example of. That's when, you know, one factor will affect how another factor performs. You know, the optimum temperature depends on pH or vice versa. But a 3-factor interaction is where you have one of those 2-factor interactions, but the fundamental nature of that 2-factor interaction changes as you change the level of a 3rd factor. So I'll show you kind of what I mean here. So in this system, we found that when we had a low plasmid copy number, and you left it for a decent amount of time, then we had this opportunity to substantially improve our yield by having also a low temperature. All the other sort of conditions that are represented on here are roughly equal. They're all about 0.4 units per mil. So it's, we kind of got 0.4 units per mil, but if we actually do this kind of long, slow expression, so high amount of time, low temperature, low plasmid copy number, we can almost double that. So that's a really nice opportunity. But then when we have a high plasmid copy number and we kind of do the opposite, so we give it, you know, we're basically trying to ramp it all as fast as we possibly can, high temperature, low time, high plasmid copy number, you know, ramp everything up to 11 and hope it works. Well, that's actually where the whole thing crashes. You know, this is 0 here. So the whole process just falls off a cliff. And we refer to these as design cliffs within the company, and you kind of see them everywhere. And just anecdotally, it's the kind of thing, you know, we're all used to that sort of experience of, you know, running a PCR one day and then it fails the next, and we're not entirely sure why. And it's my conviction that often this is due to basically there are parameters within these things which we don't have full control over, and actually they're tipping us over the edge of one of these cliffs in multidimensional space. And so by doing things in a multifactorial manner and really understanding the space we're in, we can understand where the system is robust, and so we can avoid these pitfalls. and get much higher reproducibility of the processes that we're running, whether that's, lab processes that we want to run as just a course of our R&D, or maybe biological processes where we will actually want to produce something at scale. The principle holds irrespective. And in this case, it's very obvious how we avoid that design cliff. We just make sure that we use strains where the plasmid copy number is low, and we've locked ourselves into a robust part of design space. And there's just no way that we could have got this insight without using this kind of methodology. It's only by mapping out the three-dimensional space in the way that design of experiments gives us the opportunity to, that we had this opportunity to make sure that we weren't going where the whole system could collapse, and we were going where it gave us the opportunity to actually produce substantially more than we otherwise would have been able to. So then finally, the last stage is, okay, well, we've identified some really critical factors, and we'll run quite a dense experiment where we're looking at 5 levels of each one of those factors and modeling a more sophisticated response surface in there. And once again, by following the modeling, we found that we could go up once more to levels that are even higher than we'd seen previously. And, I'll remind you that, we were actually quite happy in the first week, and then to be able to, get such dramatic improvements in such a short space of time was really a testament to the power of this method. But as we've been going along, then what have we been doing? We've been designing experiments, running those experiments, building models, and then designing another experiment off the back of that previous one. And so it's kind of almost as a confirmation of the previous model by running another experiment, which is, kind of where that model would predict things should get better. And then if things have got better, then you can see that that model is at least partly useful. So it's a fairly pragmatic way. We don't know for certain that the models are exactly correct. And, frankly, we don't really care all that much, so long as actually it's giving us enough insight to make really substantial progress. But when we get to the end here, are we confident about, these highest levels of expression or not? Well, probably not, actually. We need to make sure that we can get consistent expression there. So what we do then is we can set up a design which we would predict that all of the points in that design space should give a consistently high yield. And we call that a robustness study. So that's what we did last, just as a kind of 5th step in our 4-step experiment, just to confirm that these things were, we can get consistently high expression. Now, when we look back at the literature precedent for this particular process of making this particular enzyme in E. coli, we found that actually, one of these design points was based on it. It was kind of one of these that you can barely see the point against. And what this represented was a 200-fold yield improvement from where we'd started, and consistently getting expression levels above that. It wasn't just a one-off. So hopefully this sort of whistle-stop tour through this case study has given you an example as to the power of this kind of system to unpick complex biology to get where you need to go in a rapid and robust manner. And, I've given one example here, and James in the next webinar will give you a lot of other examples, but just a very quick whistle-stop tour through those. We ourselves or our customers have done these kind of design of experiments in quite a transformational way for all sorts of different applications, whether that's in bioprocess, assay optimization, so optimizing sort of molecular biology or synthetic biology processes like construct assembly, stitching together DNA, transformation, transfection, primary cell differentiation and expansion. So, you might have thought in my case study, oh, well, that's E. coli, that's easy. True, it's, a lot easier than working with primary cells, but that's almost even more reason why you should be using these kind of methods as your system gets even more complex. And we've also used it internally just for making sure that liquid handling is doing exactly what it's supposed to. So when we have automated liquid handlers moving, 5 microliters from here to here, we've used a design of experiments approaches to make sure that that's done with real precision and accuracy. So it can actually be used for a very, very broad range of different purposes. And there's something I really want to kind of stress as well, though, and this is, part of my job, I think, in this webinar is to try and convince you of how powerful DOE can be. And hopefully I've already gone some way to doing that with my very brief case study. But I also want to kind of give you an idea that it's actually pretty approachable as well. It's a pretty resilient method. It's not something which, if you get a few things wrong, then it's all just going to fall apart. When I first did a DOE, and actually when a lot of people in the company first did their DOEs, you just kind of get a very basic knowledge of how to do it. I did it by just getting a kind of DOE for Dummies type book and a free license to a bit of stats software, a free trial license. And I just cracked straight on, and it wasn't, I didn't really know anything about DOE at the time, but I just followed some of the basic principles, and already it gave me dramatically better results than I could ever have imagined from doing one factor at a time. And really, this is the critical thing, I think, is, you know, you'll notice that a lot of people who've used DOE get very evangelical about it, myself included, very passionate about it. And that's because, you know, once you've seen the results that it can give you, then you kind of get converted in this way and you just want to tell the world about it. And so if you've got doubts about it or can't quite see how it might apply to your system, I'd very much encourage you just to give it a crack. Just give it a go, see what it might do, and see how it might affect your experimentation and the insight that you can get from each experiment. You know, the first time you do it, it'll take an investment of time and effort, but then I'd argue that every experiment does, and not all experiments give you such transformational insight. Having said that, if you really want to push it, as we did, you know, the sort of history of Synthace can be viewed as, you know, us going from, you know, pretty complex experiments, but doable, to ever trying to expand our ambition of what is possible to address with this kind of experimentation. And we found that actually, beyond a certain point, it does get really, really tough. So I'm talking as you increase the number of factors you're looking at, as you increase the number of levels that you might want to look at each one of those factors with. You know, sometimes you look at a system and you're just not comfortable just choosing 2 levels for any given factor, and you want to fill that design space in a bit. And then DOE can get super complex. It can get very challenging indeed. And so just in the last sort of couple of minutes, what I want to do is show you how we went on that journey ourselves as Synthace, and kind of building the tools that we made for doing these kind of more ambitious DOEs that I think are almost an inevitable progression from starting off with simpler ones in the first place. And you know, once you're convinced of this method, then you kind of want to apply it to everything, and you want to apply it in every more powerful ways if you're anything like us anyway. So we essentially what we have is software which allows the execution of the planning and execution of DOEs an awful lot easier than you'd otherwise be able to do. And what it looks like is something a bit like this. So on the left-hand side, we've got a snippet of a DOE design file. And then on the right-hand side, this is the representation of the DOE workflow within Synthace. Effectively, what this is doing is it's taking in the input of the DOE design file, and then it's working out every single liquid handling step which is required to then satisfy the conditions of each one of the runs that has been specified by that DOE design file. So it's working out a huge amount of logistics and frankly pipetting that's required to actually satisfy that really complex design space. And you know, then it gives you a preview of what the system has worked out in terms of exactly how that experiment will run. So hopefully you can see this video in some level of fluidity, but suffice to say, you know, every single one of these runs is completely different from all of the others. We have 12 different inputs, a very large number of different liquid handling steps, all carried out automatically and off the plans that the Synthace software automatically generated. And so it means that we have this opportunity to run experiments that would just previously have been impossible. So that's just to give you a little bit of a glimpse as to what's possible when you then combine DOE with software for running experiments and automation as well. But that's not the main point of this webinar. The main point was to give you an idea as to the power of DOE to look into biological systems. And so very, very briefly then, to sum up, we started with kind of the nature of biology and the nature of experiments. I hope I wouldn't have to spend too much time convincing you that biology as an emergent system, it's all about interactions. You know, the product of 4 billion years of evolution, which has had absolutely no obligation to be understandable, absolutely no obligation to adhere to any kind of reductionist rules. Biology is wonderfully complex, but we need ways of addressing that complexity. And DOE can reveal those interactions. Without multifactorial methods, you're blind to them. So you're blind to one of the fundamental features of biology as biologists trying to investigate the system. And the good news is that DOE is completely within reach. Takes a bit of work to work out, what you're doing with it, but fundamentally, it's just a reasonably complex experiment that your first DOE that you try. And actually, I'm sure that all of you or most of you have done pretty complex experiments before. It is not dramatically different. It's not dramatically mysterious as to how it works. And hopefully this is giving you a bit of a sense of its kind of pragmatism, but still the power that you can get if you take this kind of approach. So before we go into Q&A, I just want to make some acknowledgements. As I say, DOE's been at the heart of Synthace pretty much since our inception. And so this is kind of thanking all at Synthace, really, both now and over the years. But particularly for the work presented, I wanted to thank Dr. Laurent Malivert, who actually carried out the lab work in those early days of Synthace. Sid, Michael Sadowski, who has, since the very beginnings of Synthace, been at the heart of our experimental design strategies and interpretation and modeling. And also my co-founder, Chris Grant, who again has had an instrumental role to play in both thinking about DOE, but also how we could actually run DOEs within our software. And also, genuinely, we've seen some really, really exciting stuff happening at our clients recently using these kind of tools. And it's when you get the combination of some really ambitious biologists who really, really know their stuff about the system they're working on, who can design these fantastic experiments, which can give them further insight into that system, combined with the tools that we're giving. And it's giving some truly inspirational results that hopefully in a day, not in the not too distant future, we'll be able to share those with you as well. But I just wanted to give them a shout out while we're here. And with that, I think we can move to some Q&A.